All right, everybody. It is now 1 o'clock. We'll get started. Appreciate everybody joining. My name is Greg Bryan. I'm a commercial manager at EnviroZyme. Matt Livingston is our global technical director. And this discussion will be on optimizing refinery wastewater treatment. And if anybody has any questions, please use the Q&A. There won't be any audio or anything like that. So at the end of this presentation, we will have a Q&A session, and I will read the questions aloud and potentially respond to any questions as they come in. So thank you very much. And on that note, Matt Livingston, take it over.
All right. Thanks, Greg. We will just get started because we have an hour. So first of all, thanks everybody for your time and attention this afternoon. I know a fair amount of people on the line are partners and people we know, also some folks that we don't know. So thank you. Welcome to this presentation this afternoon. At EnviroZyme, we look at a situation around wastewater treatment using a lens of biology. So we prioritize biology in our approach to troubleshooting wastewater treatment plants, in particular refineries. Refinery wastewater is a complex mixture of a lot of different types of substrates, hydrocarbons, phenols, sulfides, but lends itself well for biological solutions. So those are some of the things we're going to talk about today. Let's see. Into the agenda, since some of you are unfamiliar with EnviroZyme, we'll do a brief introduction to the company and about our technology platform, Bioaugmentation, and then we'll get into some other things around refineries and some of our other troubleshooting approaches. EnviroZyme is the world leader in bioaugmentation. We're a subsidiary of the Betco Corporation. We're headquartered in Bowling Green, Ohio, where we have our administrative offices and our production. On the right-hand side, you see a picture of our production facility from about a year ago. That facility is now twice as large. We're opening up new production sometime this quarter, third quarter, which will be almost doubling our capacity. So we're excited about that. We're a GMP, ISO certified, FDA-registered facility. And here we ferment, blend, and package custom and off-the-shelf microbial solutions. We have local sales support and technical support covering North America, Latin America, Europe, and Asia Pacific. And we truly have a global presence. We work with both local partners and global partners to support end users. So we don't sell directly to customers. We work through distribution and channel partners. And again, it's a combination of global partners and also with local partners around the world. EnviroZyme came to be. Betco Corporation acquired Biosystems International in 2009 and then rebranded that company into EnviroZyme. Some more recent acquisition activity in 2023, EnviroZyme acquired the wastewater division from Novozymes. That's where I spent over 20 years of my career. And then more recently, at the end of 2024, acquired the wastewater activities from BioWish. So rolling those companies up to make us really the largest player in this field. And as I said, our headquarters in Bowling Green, Ohio. It's about an hour or so south of Detroit in the United States here. Okay, what we do with our products and what we do at our production facility here is really ferment bacteria and produce bacteria. We start by finding the right organisms, naturally occurring organisms that occur naturally. We look at environments where we think we'll find the right type of bacteria to be successful in a wastewater treatment plant. Sometimes it's from a wastewater stream. Sometimes it's from a soil. A lot of the microbes that you see in wastewater treatment systems are native soil microorganisms. But we find those organisms with specific either degradation capabilities, so whether they can break down certain types of targets, whether it's hydrocarbons or ammonia or different substrates, or for organisms that will survive under certain conditions to make sure that they're going to survive in the application. When we put it there, we get those organisms, we test it out. When we confirm that they're the right strains that we want to bring into our production, we have a scale-up procedure where we first grow them on a petri dish or in a liquid medium, put it into a smaller flask, grow it into a seed fermenter, doing this all one strain at a time. It's a very sterile process. We go up to a main fermenter, and then we concentrate the material. Some of the strains will spray dry it. Some we set aside as pure cultures, or we'll mix them together. And at the end of the process, generally come out mostly in wastewater treatment with dry products that are in a powder form in a water-soluble packet that can be added to a wastewater treatment plant. We also have some solid products and some liquid finished goods, depending on the type of bacteria and the application. But that's our production process. When we talk about bioaugmentation, we're talking about the process of enhancing the indigenous bacterial population. So it's not taking over a population, it's really adding or enhancing that indigenous population with the addition of concentrated, naturally occurring bacterial strains with either specific capabilities or specific growth abilities. We kind of look at four basic applications with bioaugmentation. The first being a startup. And the idea there is that when you bring a wastewater treatment system online, you want to bring it up as soon as possible. So getting bacteria in there that are going to start up quickly and get the biomass built up to a certain level so the treatment is at high efficiency as soon as possible. The second application is system efficiency. So that's looking at improving the overall plant efficiency. A lot of times you'll build a factory, say in the 1960s, 1970s. Over time, that factory will expand, but the wastewater treatment plant stays the same. In this application, either undersized plant or some other challenges, trying to boost the efficiency of that system so that you're meeting permit or meeting the goals of that wastewater treatment plant, whether it's settling, BOD, nitrification. Stability, and I would say in refining, stability is one of the key applications in a lot of plants where you have stressors coming into a wastewater treatment plant. Sometimes that your performance might dip a little bit. The idea with a regular dose of bioaugmentation would be to make the plant a little more stable, handling some of those variations on the influence stream coming in. And then if the plant does have an upset situation, bioaugmentation can be used to get that upset recovery over with as soon as possible to get the plant, similar to a startup, getting it back up to high efficiency as soon as possible. Again, in a refinery application, especially around nitrification that we're going to talk about, that's a key application for bioaugmentation. Okay, so that's a little bit about the company and our platform. We'll talk next on refinery wastewater and a little bit on why this is a good application for our technology. When it gets down into crude oil, of course, is the feedstock at a refinery. It's a very complex mixture of hydrocarbons and a lot of other components, metals, other minerals, other contaminants, and that varies a lot by deposit and location around the world. So it's a very complex supply chain, getting crudes, shipping crudes from all over the world, processing in different places. One of the things, and I would say, not just immediately, but over the last 15, 20 years, is the just seeing a lot more sour crudes and heavy crudes in the market. I remember in the 2000s, it was maybe more of a novelty to have refineries bringing in sour crudes, and that was a big deal because it was very difficult. Sour crudes are now cheaper and more abundant, and the technology to process sour crudes is more readily available, and you can do it at a profitable way. So that's the predominant crude on the market, and in the supply chain, we're just seeing a lot more sour crudes and heavier crudes. And crude does have an impact on wastewater treatment, especially switching from one crude slate to another. Again, on the sour crudes, anything more than 0.5% sulfur to up to 5% is a sour crude. And that's important because the technology has to be there to remove sulfur from the process and also be able to handle amines. Amines are used to sequester sulfur and sulfides during shipment because sulfur and sulfides are corrosive material. So a lot of amines can be used to sequester that, and that can be really damaging and hard to treat from the wastewater treatment perspective. So as I mentioned, sour crudes are very common today in the market, maybe more than it has been previously. And really, most refineries are treating sour crude. A lot of these, this was just from a report I picked up, but a lot of these are customers, and we know a lot of these facilities that are treating sour crude. And a lot of the issues that they have are a result of treating sour crude. Even though it's profitable from the refinery standpoint, refineries have spent a lot of time and a lot of investment on buffering their plants, buffering the wastewater treatment system, so that they can handle some of the challenges from sour crudes, although it still can create problems. Not to get into the refinery process too much, but you have different processes around refineries. All refineries are a little bit different, but these are the major refinery units, and they contribute to wastewater in different ways. Sometimes, especially when we're dealing with a unique toxicity situation, sometimes it's helpful to understand what's causing the toxicity and potentially where that's coming from upstream in the refining process. So it's just a little footnote here of what compounds generally come from what different processes, and sometimes it's helpful to backtrack upstream. And again, while toxic at higher concentrations, most of these compounds are degradable and can be managed through biological treatment. So that's either through operations at the wastewater treatment plant or through different technologies, including biological products like we're going to talk about. And then going downstream, pre-treatment is really critical. Again, it's kind of the last defense for a wastewater treatment plant. We have an API separator or a DAF or equalization. Sometimes the order of those three can be switched around. But in a place where pre-treatment is neglected, they're going to have a lot more biological issues. So it's really important to get the pre-treatment right. And folks there, don't overlook that. It's worth optimizing and maintaining. And again, it's kind of the last line of defense for the wastewater treatment plant. When we get into biological treatment, like very simply at the most basic form, like the primary goals of wastewater treatment is for bacteria to remove pollutants from the water. You know that, and then to form a dense flock that can settle out and be removed. So in this case, you see a real nice flock in the middle. On the right-hand side, you see a nice settler with very clear water on the top and a very clear line of where the sludge had settled. That's a really nice looking sample. In a refinery, sometimes that's going to be hard to find a sample that looks that good. In a refinery, there are a lot of growth pressures that impact the flock and a lot of stress that causes challenges to that process, to getting a good flock, to getting good settling. We talked about toxins a little bit, phenols and sulfides, amines can come in. And again, if it's not regulated, if it comes in as a slug dose, that can really impact the biomass, cause stress to the biomass, and cause some treatment issues. Sludge retention time, also a key growth pressure in determining how well your biomass is going to work. And that's going to be different for different systems and different wastewater treatment plants, depending on the loading type of system that they have. Often in refining, sometimes we come across customers running very long sludge ages. That can be helpful if they have toxicity and need more time for the biomass to get things done, like nitrification, but can also be challenging because some things, you get into some complications when you run your sludge age or sludge retention time too long. You have a lot of varying pHs in refinery. You're dealing with caustics, dealing with acids, and sometimes it's uncontrollable and you don't know what's coming down into your plant. You can have a high pH, low pH, but those spikes, again, can just be very disruptive to the biomass. The substrates that come in, again, if you're dealing with sweet crudes, you don't have as many contaminants, don't have as many issues. But if you're dealing with sour crudes, substrates can vary depending on the crude slate. Again, if you're changing crude slate, that's a big stress to wastewater treatment plant. And a lot of times that's one of the first questions when we see a big shift in kind of operating performance. that's often a telltale sign of a change in crude. Dissolved oxygen is also a growth pressure. Sometimes there's limitations in terms of the infrastructure that's there. And also sometimes in refineries where you have a lot of contaminants or inert material or EPS that we're going to talk about, that can impact oxygen transfer efficiency. So even though you might have a good DO reading, 2.5, 3, 3.5, that oxygen might not be able to penetrate into the flock because of inert material. So that's also can be a challenge. Temperature, especially on the high side in refineries. I was in South Korea last month, went to 4 different refineries, and all of them are really stressed around temperature. One of them was up in the 40, 41, 42 degree range, and really seeing some impact on nitrification and some other processes and settling and flocculation. So high temperature can be an issue. We talked about inert material and just competition among different bacterial types, whether we're talking bacterial filaments or other things, just having the right biology in place. Competition from other bacteria can cause an issue. And a lot of this, is determined around decisions that operators make and operations management makes. And that's key to really managing the growth pressures is the decision making around how the plant's operated. Okay, so next, want to talk a little bit about, when we get called in or when our partners get called in around bioaugmentation and looking for potential applications, how do we approach a plant? And I think the first level is understanding what the issue is, but then doing a deep dive into the data, looking at log sheets, trying to get information from the customer. It's really important to do that. Sometimes anecdotal, just describing a problem isn't really enough. Going on site, understanding the design of the plant, and then asking questions. Is the right data being collected? Does the data that you are seeing in the data sheet, does that describe the problem that the customer is observing? Can you find where those issues are in the data? Or is it something that's really hard to find in the data and they're experiencing some challenges and it's hard to nail down? But it's really that first level is really a combination of looking at the data, getting on site, talking with the customer, understanding the system, and seeing if the chemical parameters, physical parameters promote the right kind of biological activity that they want to have happening, whether it's BOD, settling, nitrification. That's kind of the first level of how we operate. The next is kind of on a microscopic level. So doing a microscopic examination, and we could do a full day training on microscopic examination. We work very closely with the Leach Microbial Consulting. Steve Leach is a former colleague of ours. We do a lot of work with him, and looking at the biomass under a microscope can be very helpful in supplementing kind of your understanding of the data. You're looking at the run sheets on the last one to see if the parameters are right, but then looking at the biomass under a microscope can really confirm that. So from the flock standpoint, what's the size of the flock? Is it a large flock? Is it dense enough to settle? Is it a compact flock? Are there enough filaments to hold the flock together? Are there too many filaments that the flock's going to be hindered in terms of settling? A lot of times in refining, you get kind of a weak, small flock, again, just from the stress that's there. But it's helpful to look at it under a microscope to get an understanding. When we talk about bacterial filaments, sometimes filaments are helpful. They can be kind of a backbone to a flock. They can, provide structure. But if there's too many filaments and if the filaments are growing out of the flock, that can cause challenges with settling. So in the clarifier, it takes too long to settle down to the bottom, so you don't get a very concentrated return. You can lose TSS over the weirs if it doesn't settle fast enough. Also, it makes challenges with at the bottom of the clarifier where it doesn't, it stays too separated. It's not very dense at the bottom, so compaction is what I'm trying to get at. So you don't have a very compact return mixed liquor. you can identify the different filaments, and a lot of filaments will grow under certain types of conditions. So there's low nutrient filaments associated with low dissolved oxygen or a low food to mass ratio. So again, this is a great confirmation when looking at run sheets or looking at the plant data. If there are a lot of filaments, confirming the reason and trying to understand the reason in the data on why you have certain filaments there. Looking at higher life forms, so these are larger non-bacterial, but often single cellular organisms or multicellular organisms that are large enough to see under a microscope. The organism here, a suctorian, is, if you see that around a plant and you're nitrifying, that's a really good sign. You see these suctoria often in plants that are nitrifying. If you're used to seeing that at a plant and all of a sudden you do a couple micro exams and you stop seeing this, that could be an issue that maybe there's something on the horizon coming in, kind of a canary in the coal mine situation where that can also kind of give you some clues about what's happening in your system. And again, looking at the bulk water and understanding, is the bulk water clean? Is there a lot of inert material? Free bacteria in the bulk water can really give a good sense to the health of the system. The next layer is something that we see more and more, is doing bacterial analysis, a DNA analysis on the community, the microbial community. I would say 5 years ago, 10 years ago, when this technology first started coming out, it took a long time, it was expensive, and the output was kind of complicated. You get a long list of 100 different types of bacteria or more, and it was a little bit hard to make sense of that data. Generally, the data gets down to the genus level. If you're a biologist, you have kingdom, phylum, class, order, family, genus, species. So not down to the species level. Usually it's to the genus, and there are often hundreds. We work with a company with our partner Aster Bio, and one of the really nice things that they're able to do is work with databases that have now been running for 10, 15 longer, that have a lot of information around different types of bacteria that are found in different wastewater treatment plants, and an understanding of, are these ammonia oxidizing bacteria, nitrogen oxidizing bacteria, and I'm going across that row on the top and give a percentage of what percent of the bacteria in that community have the capability of that based on the genes that you see. So nitrogen reducing bacteria, sulfur oxidizing bacteria, sulfate reducing bacteria. So if you're in a system where you see a lot of hydrogen sulfide, you can look to see if you have a lot of SRBs, if that's contributing to the hydrogen sulfide. Phosphate accumulating organisms, all the way down to different types of filamentous bacteria or foaming bacteria or bacteria that are commonly associated with EPS. And that's a really helpful way, just a short 30-second snapshot, to get a sense of what the bacterial community not necessarily is doing, but what it's capable of, because we're looking at the genes that are in those organisms. It's really helpful to do this on a regular basis when your plant is running well, so that when there is an upset, you can compare kind of where your plant's at versus where it normally is. And that can sometimes provide, again, a good window into treatment opportunities. So now combining kind of bioaugmentation in a refinery setting, again, where we see kind of common applications, again, plant startup, but more likely it's, say, a recommissioning after a turnaround or something like that, where they've stopped production or stopped flow for a certain amount of time. In the system efficiency, it's around overcoming design issues or temperature issues, or if you're having BOD, ammonia, TSS, certain hydrocarbons are leaking through the plant, or phenols, looking at products specifically for those applications. Again, from a stability standpoint, again, probably one of the more common applications that we have is just giving the system a little more resilience against variability of feed from changing food slates or toxic material that's coming in. And then a recovery situation is often around nitrification or recovery from some sort of toxic washout. I will say, and I'll repeat it later also, when you're dealing with a toxic upset or a toxic nitrification situation, you want to make sure that that toxic material is washed out of the plant before you start adding product again, or you're just going to have the same situation if it's a toxic environment. So there are things you can do, and we'll talk a little bit about that, about just making sure the plant is in the right condition, I would say ready for a bioaugmentation solution if you're going to go that route. Okay, so specifically around some wastewater treatment, refinery wastewater challenges. Just going to talk about a couple things today, again, because we're limited on time and the scope here. But I think these are things that should have a broad relevance. The first is EPS, and EPS stands for extracellular polymeric substances. And we'll talk a little bit about some of the challenges and control strategy for EPS. This is the most common biological problem globally with refineries that process sour crude. I know when I was in South Korea last month, all the refineries had sour crudes and they were all experiencing issues around EPS. Our partners that work with refineries that are in the Gulf Coast talk a lot about EPS, especially with crudes, sour crudes coming up from Latin America or Venezuela. Some refineries in the Midwest in the US often dealing with crudes coming from Northwest Canada. Again, sour crudes, heavy crudes, often contributing to EPS problems. So this is really the number one issue. And again, 20 years ago, didn't see as much of it, but with the proliferation of sour crudes, we're seeing more and more. And what it is flocks with large amount of this material, they hold on and bind a lot of water and a lot of fluid and a lot of slime. So EPS consists of polysaccharides, proteins, lipids, DNA, and causes a lot of issues around settling and dewatering. EPS is different for different bacteria. There are some bacteria that are more well known for the type of EPS and EPS that they make in a wastewater treatment, but it varies depending on the environment. And slime and EPS can be visible under microscopy using either an India ink stain where the India ink cannot penetrate into that. So the image on the right-hand side, you see normally a normal flock, India ink particles would penetrate into the flock particle, but in this case they don't. And that's just kind of almost a jelly-like mixture that's really hard to settle and dewater. So that would be what it looks like under the India ink. There are two types of EPS that we see. One type is called tightly bound, and tightly bound is EPS that's very close to the cell, and it almost looks like a capsule around each individual bacterial cell. We see that caused by readily degradable substrates. So when you have a high FM situation, high food to mass ratios, sometimes a nutrient deficiency. And what happens is the bacteria is really taking food that's available in the environment, not digesting it all the way, but kind of storing it up and almost putting it in a backpack and saving it for later in this form of EPS. I'm going to take one more click and kind of blow that area up. So you can see that part of the flock looks different from the rest of the flock. And each of those bacterial cells almost seems like it has a little capsule around it. And that's the EPS that we're talking about. Again, it's harder for DO to get into that flock, and it's harder for that to dewater, and it doesn't settle as fast. Sometimes that can almost look like a foam or a scum on top of a clarifier or on top of the treatment, on the top of the aeration basin when you have so much tightly bound EPS. Loosely bound EPS is the one that we see more of, especially with regards to refineries that are treating sour crudes. Almost looks like a slime coming out. I'm going to press it one more time so you can see kind of a blow up of that area. It's caused by stress. So a lot of different factors can cause stress, not just one. But what's happening here, again, the bacteria are storing up reserves for later because they may be in a toxic situation where their cellular machinery isn't going to be running so well. So they'll save that up till a later time when they can use it, or a protection from toxins. So there's kind of a protective layer in there, or some sort of protection from an environmental change. So you can see in this matrix, the bacteria are pretty far apart from each other. They don't grow so closely together, but in that area, it's all this kind of polysaccharide extracellular material. Again, that's very difficult to dewater and very difficult to settle. You generally see that with a longer sludge age, maybe 10 days, but 20 days or longer is where you often see kind of buildup of loosely bound EPS. And again, it can come from a variety of different stress type situations. And this is the type of EPS that we see more commonly in refineries. Some of the issues around EPS, poor settling and dewatering. So the chemical use is going to go up. Sludge disposal cost is going to go up because you can't get your cake as dry. It can also impact nitrification. So nitrifying bacteria really need to survive on a flock particle. They don't do very well just floating out in the bulk water because it takes them a long time to reproduce. If they're just in the bulk water and they don't settle out, they'll just wash out over the weir and you'll lose nitrification. And nitrifiers don't accumulate very well when there's a lot of EPS, so they don't really join that EPS flock. They like to be in a more dense flock. So sometimes you'll lose nitrification, or you can also have issues around ammonia violations or possible wet testing because your ammonia spikes and then that starts to impact toxicity around your wet testing. You can get a lot of foam and scum on the DAF or aeration basin or clarifiers. That can lead to a TSS violation or just, be expensive to remove it, right? It just costs money to remove the foam and scum. And then again, other issues around oxygen transfer for nitrification, BOD removal, COD removal. When you don't have as good oxygen transfer efficiency, which can be caused by EPS kind of blocking dissolved oxygen by getting into the foam, getting into the flock, you can have ammonia violations, BOD, COD treatment can worsen, and also issues around wet testing. So several issues, again, not just from having the EPS there, but that EPS causes other issues in your treatment. Kind of a list of possible causes for EPS. There's a lot of things on here, but this is still probably a limited list. From the loading and feed standpoint, from feed variability, any increase in organic loading, increase in inorganic loading, crude change, or an unknown chemical dump, because that happens. Nutrient or chemical imbalances from pH swing or low nutrients coming in. If a plant's running a chemical trial and that doesn't go, that can also impact wastewater treatment. And that's one of the areas sometimes where we see EPS when we're not expecting it just from the chemical trial. Temperature, seasonal shifts from one season to another, whether it's getting warmer or colder, that shift is enough to cause stress on the biomass. And then operational disturbances from either process shutdown or equipment, feed pump issues, operational changes, aeration or mixing issues. All these can cause stress, and stress causes EPS. So some of the approach that we would look at EnviroZyme with our technology around bioaugmentation, around treating EPS. So EPS can only be removed by wasting. It's not going to just dissolve or go away. It really needs to be wasted out of the system. So often the recommendation is to increase wasting to get the EPS out of the system. We would recommend bioaugmentation to build back a healthy biomass. In the case of tightly bound EPS, where you have kind of a readily degradable shock load, adding bioaugmentation on a regular basis can help decrease that shock load from the material coming in. And then from an operational standpoint, if the plant has the ability to introduce step feeding, that is taking the influent and moving it not only at one spot in the aeration basin, but maybe passing it around to distribute that degradable BOD load so that it's not just in a high FM situation in that one part of the plant that can cause issues. If you spread it out, sometimes that can certainly help the plant accommodate that kind of loading. Similar with loosely bound EPS, it can only be removed by wasting. So the first thing you want to do is identify the causative factor, look through the kind of growth pressures, try to understand where that stress is coming from. try to make some changes to the system. Again, sometimes those changes are going to take 1 to 2 sludge ages, 2 or more sludge ages to work their way into the biomass to show those differences. But be patient, make some operational changes, and try to improve or reduce the stress. Bioaugmentation can help with that. Again, if you're wasting out your sludge, trying to build back with a healthy biomass can be helpful. Having some organisms in there that can degrade toxic compounds, phenols and amines, can reduce stress, and again lead to reduced stress on the biomass and reduced DPS. One of the products I'll highlight here is called BioRemove HC. It's really targeted around different types of hydrocarbon and different types of bacteria that are in this product to degrade a variety of hydrocarbons that we see come into refineries. Reduces violations, targets oils and grease, and can help with hydrocarbon-related filament outbreaks, and just improving the system stability overall. So a summary of EPS, again, globally, it's the most common issue that we see in refineries, leads to high chemical costs. If someone is unaware that they have these issues, it's hard to deal with it. So, trying to get samples in under the microscope is really the best way to identify that, and, get in touch with us and we can help facilitate that. And then with both tightly bound, loosely bound, you need to waste that material out, make the right changes to your plant, and try to build back a healthy biomass from the wasting. All right, the other issue I wanted to kind of dive into a little bit is nitrification. The nitrogen cycle in the environment. Nitrogen presents itself in very different forms around the environment. It goes from nitrogen gas to nitrite and nitrate to ammonia, organic nitrogen, and that cycling and those natural processes are really taken into consideration in wastewater, and it's the same processes that are happening in a wastewater treatment plant. So especially in a refinery where either you have organic nitrogens or amines coming in, bacteria can convert those more complicated molecules into strip off the ammonia and increase the ammonia in your wastewater treatment influent. And we see that often in a refinery, especially with sour crudes. Assimilation is really the bacteria that are in your system taking in nitrogen, taking in ammonia, and using it as kind of a building block for part of its growth, whether it's DNA or proteins. Bacteria need nitrogen to grow. Nitrification is where bacteria use energy in converting ammonia to nitrite and then nitrate. And this is kind of the classical ammonia oxidizing, nitrite oxidizing two-step process. And again, that's what we see in most refineries. And then denitrification is, of course, taking nitrate and converting that into nitrogen gas. But we'll focus here mostly on nitrification in this discussion. We kind of offer up a nitrification checklist where we look at pH, we look at dissolved oxygen, temperature. Alkalinity is one that's often overlooked. Again, temperature often on the high side, anything over 35 can really cause some challenges. Phosphorus is something that's often overlooked in refineries. Again, like nitrogen, phosphorus is assimilated into bacterial cells and needed for producing energy in the cell. And if there's not enough phosphorus orthophosphate in the wastewater, you'll see a decreased bacterial activity, especially around nitrification. Making sure you're removing enough BOD and COD. Sometimes if you have too much BOD, there can be competition for oxygen because nitrifying bacteria grow more slowly. They can be outcompeted if there's still too much BOD. And then sludge age, just make sure it's long enough. Often in refining, it's over 10 days. Again, over 30 days, sometimes can wind up in a situation where you might build up toxicity in your sludge. So when would you consider bioaugmentation for nitrification? I'll just hit a couple of these applications. One is where you have frequent upsets with a slow recovery. Another is where you have a history of nitrification, but you're not able to recover at all, so you're stuck there, or a plant that has no history of nitrification. And with that one, it's a little small here, but that's a yellow light. Definitely want to qualify that plant before taking any action on it. With a frequent upset and a slow recovery, again, we talked about a variety of reasons why you can get an upset. And again, if the upset is taking more than 1 or 2 weeks, it's a good opportunity to explore bioaugmentation. This is an example based on some customer data. We work closely with customers, but often don't have permission to use customer data. So this is kind of simulation, but very similar to some of the applications that we see. In a municipal plant, someone might lose nitrification and bring it back in a couple of days. In a refinery, again, just because of the matrix of what's in the water, it takes longer often for recovery. So in this case, it's about 20 days out. We would typically look at the nitrification checklist and, cutting to the chase here, looking around COD and, is it a phenol spike or something else? What we notice in this case is that the pH dropped off around somewhere between June 22nd and June 29th. We also see that in the alkalinity impacted, that it drops off. But when you get a low pH, again, for 2 days or so, going below 6, that's enough to really impact your biomass. So kind of identified the situation. That's what it looks like here in terms of when this pH spike hit and when the recovery was. With bioaugmentation, what we would tend to see is a dosing program of somewhere around 5 to 10 days, typically starting as soon as possible. We generally have this product available to ship overnight so that we can get very, a fast response on it. And in this case, you would look at an upset recovery, maybe 2 weeks, 14 days faster using bioaugmentation. Again, it's getting enough organisms in there that have that capability while the system is still kind of repairing itself. This system is going to grow back nitrifiers, and it will be, nitrifying well healthy beyond the period of time where we're adding products. A lot of time, the products is just to get it through that hump to when the system's able to repair itself around nitrification. And I realize it's about 20 of, got 2 or 3 more slides here on nitrification, and then we'll jump into some question and answer mode shortly after that. Here's an example of a plant that is unable to nitrify but has in the past. So the question becomes, what's changed? Is it a different type of crude? Are there new processes? Really jump into that question around the data with the end user and really understand what the issue is. Go through the nitrifier checklist, make sure that pans out, do a micro exam, even looking at the microbial community analysis and understanding, okay, if you're used to seeing certain types of nitrifiers, are they still there? Are they not there? Again, if the nitrifiers are not there, it's very clear that the plant's not going to be able to nitrify without getting some, A, fixing the conditions, and then B, getting the right organisms into the plant. You can do a toxicity screen with our nitrifying bacteria, and I'll give a quick example of that because we recommend it frequently. Here's an example of a plant that had a 35-day sludge age on the long side. Nitrification had slowly deteriorated, and then it was lost, and we had suspected toxicity. So setting up a benchtop test that can be done on site, pretty simple, 100% influent and mixed liquor, and then 100% mixed liquor. And we'll dose that with our nitrifying bacterial product, BioRemove AM. It contains both Nitrosomonas and Nitrobacter, ammonia oxidizing and nitrite oxidizing bacteria. And what we see here in this case is that the influent fully nitrified, the blend of mixed liquor and influent nitrified pretty well, but partially, and then the mixed liquor didn't really nitrify. And what that would suggest, and what can happen sometimes, is that there's toxicity bound up into the biomass in the flock. And that would mean you'd really, again, want to waste out some of the flock. What we recommend is the BioRemove HC to replace some of the wasted sludge and to help degrade some of those toxic compounds coming in. And then repeating that test, and only when the mixed liquor is able to nitrify, start adding BioRemove AM to bring back nitrification as soon as possible. Again, in a plant, in a situation where the plant is unable to nitrify, or if the conditions aren't right, if you go through that nitrifier checklist and you can't check everything off the list, don't add nitrifiers because the system's not not going to be able to nitrify, and that's a key misconception we want to get out of the way. So again, nitrification is probably one of the more common bioaugmentation applications that we see in refinery. In refineries, got a simple checklist for that. Microexam to look at some of the stress indicators, DNA to understand what organisms are present, toxicity screen, and again, don't add nitrifiers to the system if the system isn't ready to nitrify. So. All right, it's about 1/4 of, so what I'm going to do, just in case we had more time, I was going to briefly talk about filamentous bacteria and settling and compaction. I'm going to skip that other than, again, these are biological issues that have, in some cases, some biological solution alternatives. Feel free to reach out if this is an issue to you. Be happy to have a conversation. And then also around pin flock and just smaller flock and some of the common issues we see in refineries around settling. And then again, a long list of things in refinery wastewater treatment that we can address with bioaugmentation or other issues around, from the loading to shock loading and recalcitrant compounds coming through, toxicity, H2S control, again, a common issue with refineries doing sour crudes. And then finally, a list of some products that we have. So we talked about BioRemove AM as a concentrated nitrifying bacteria, BioRemove HC as our hydrocarbon degrader. We have a variety of other products around for sulfide control. The OdorCap 5700 has a bacteria that can biologically oxidize hydrogen sulfide, which presents some unique opportunities for controlling sulfide odors and sulfide corrosion. BioQuick 5140 is specific for startups. There's some inert material that the bacteria can live on and house on. So at the beginning of the startup, they get into the clarifier, they settle real well and can be returned back into the process and not washed out. We have some nutrients and some hard surface cleaners, and you feel free to get back in touch with us on that. Do want to mention our team in North America. We have, and if you want to get any of these contacts, this would be a good time for a screenshot or to look back on the presentation. I'm in North America as myself. Greg Bryan is our commercial manager. Des Thompson is account manager, and Joe Sloan is our VP of Wastewater of Water Environment Technologies. In Latin America, Juan Gerardo is director of sales. Vera is in Europe as a sales manager, and we have 2 folks in Asia Pacific, Mike McWilliam, who's director of sales out of Japan, and Bibek Malik, who is in India, is our sales manager there. So now it's time for Q&A, and thank you for paying attention, and we got through this in a good amount of time, so I will take a breather, and Greg, you can let me know if any questions have come in.
Yeah, Matt, you can hear me okay, right?
I can hear you fine.
All right, we have several questions that came in. The first one is, what kind of incoming BOD do you see in refinery systems? I'm coming from the paper industry, and they range from 300 to 1,000, all the way up to 3,000 to 4,000 milligrams per liter, and some of the mills for aerobic treatment plants. So what's the loading typically that you see in refineries?
Yeah, generally lower than that. Generally not seeing up in the thousands, in the low hundreds. Yep, is what we're seeing in that area. It's not in that same super high concentration that you're describing there in pulp and paper. That's a good question, but it varies. It varies from refinery to refinery. But my recollection is that it's in probably more commonly in the hundreds than it would be in there, and then the COD maybe often double the BOD. Again, okay, depending on the processes and the efficiency of the production.
Gotcha. Okay. Another question. Filamentous formation is common in stressed environments. Where do you see the difference between EPS production versus a rise in filamentous bacteria?
Yeah.
Good question.
That's a great question, and really the answer, I would say, really comes down to the microscope. So it's hard to say what's going to favor what, because again, I'm going to go back a couple slides to this one here, where some of the common causes for filamentous bulking, these are all stressed conditions, and a plant and the biomass that's there in some cases may result in a filamentous bloom where the filaments really get the advantage and take over and start to dominate. Or it may be that the system responds by stress and producing more EPS. So it's under stress, under these conditions. Same conditions generate both of those. But I would say it's tough to say, but I guess it doesn't really matter. It comes down to identifying what the issue is and then trying to take steps to control the issue. But again, stress is going to contribute to both of those. And I guess it really comes down to the specific conditions at the plant that are going to determine how that manifests itself.
Okay. And another question kind of related that I'll get to first. Can you see EPS or any indicators of that in a DNA test?
Yes. So there are some, especially on the tightly bound EPS, there are some bacteria that are more commonly associated. I think Zooglea was one that, 20 years ago in the manuals, it was all Zooglea. Now I think there are 4 or 5 other genus of bacteria that we look at that can also be cause for tightly bound EPS. Loosely bound EPS, I think that's also the case. And again, if I go back to the slide, there is in the DNA testing, there is a category that they put out for EPS here. So if you go down that list, there's going to be, and what that's going to indicate is bacteria that have the capability or have been identified with that, with the ability to form a lot of EPS. Doesn't necessarily mean that it's happening. Again, so a DNA screen is going to be confirmation that, yeah, you're seeing that. You might have a really high percentage of bacteria that are capable of EPS, but there's not a lot of stress on the system. They might not be generating EPS. So again, good to understand, but I would say the proof of that really comes down to the microscope and understanding what's being generated at the plant. And then, as you do that, and if you do DNA testing, you'll get a correlation around, okay, what does that look like when you see more EPS? Where does that number go up? And maybe you'll have a better sense of understanding that.
Okay, great. Next question coming in. Is it possible to have treatment in the desalter effluent to sewer in crude units to remove hydrocarbons such as benzene before it reaches our effluent area?
Yeah. I think the question there is going to come down to toxicity. I don't have a good answer. I don't know anybody doing that specifically. I think part of the challenge is just you don't have a lot of dilution from that area, so you have a lot of high strength contaminants coming out of that area. If you wanted to try some sort of like selector or some sort of treatment system, you'd have to think about a way to build in retention time. Treating it in wastewater, treating it upstream, and again, it's really understanding is that suitable upstream. I think in a lot of times you're going to have too much toxicity to start that treatment, at least from a biological perspective.
Okay. Next question. In which case do you advise to apply Bioremove HC, and in which case refinery and recalcitrant organics?
Yeah. So, okay, that's a very specific question from a customer coming in. I think there are some similarities between the products. Typically, we would lead with the hydrocarbon product because it's more specific around refining type applications with hydrocarbons. There's another product that we have called recalcitrant and organics that maybe has a wider range of products that can also be used in paper mills and refineries and other places, but maybe less specific for hydrocarbons. So typically in a refinery, we would lead with that, but it's also a viable product for refinery application. But that would be how I would describe the difference, just that the HC is a little more focused for hydrocarbons in a refinery setting where recalcitrant and organics is more broad for industrial type applications.
On that note, would they be able to do bench testing to see which one performed better?
Yeah. You could do that. You could do bench testing. when if you get to that stage, feel free to reach out to myself or any of our regional sales folks or our channel partners, because getting into the setup sometimes takes some good planning and understanding whether the lab has the right kind of setup to do it. Sometimes you can see things in benchtop tests that work pretty well, but you got to be set up the right way. So we can help kind of qualify whether someone's in a good position to do that or not.
Then on that note too, we're able to help you guys review data that you may have and help you troubleshoot, what may be going on in the system, just to give you another set of eyes to take a look at your data. We've got a couple more questions that came in. We've got about 5 minutes left. How much COD can be treated in the refinery effluent? Can 32,000 ppm of COD be treated?
Yeah, that's a good question. 32,000 ppm. That's a high amount of COD. Again, that's the kind of thing where you might want to take that offline and do some treatability testing. Say, do you want to handle that all at once? Is that something you possibly could dilute down? Understanding what else is in that matrix of COD, right? Is it all phenol, right? Do you have 500 ppm of phenol in there, or a lot? So it's kind of, it depends, yeah. But challenging situation.
You may want to go upstream and take a look at primary treatment. Are you getting carryover? Is that why that number is so high? So it's always also good to take a look at what's happening upstream or what is being spilled, and try to eliminate that source.
Yeah, and whether that's a side stream we're talking about, or a specific stream that you're trying to treat by itself, or whether that's obviously not combined with the influent, but whatever that's combined with, would be helpful to know. Yeah.
Yeah. All right. Well, we still have 3 minutes left. A few more questions here. Where is the best feed point for the BioRemove HC? And how long will it take to proliferate?
Yeah, typically we're going to add that to the inlet, as close to the inlet to the aeration basin as possible. So the activation point, some of the bacteria will start working immediately. We have a variety of different types of bacteria in our products that I didn't get into. We have some bacteria that are called vegetative bacteria, and those are bacteria that are alive, but in our dry form, and when we ship it, just in a very slow metabolic state, but will wake up pretty quick when they get into a wastewater situation. Some of the types of bacteria that we have are called spore-forming bacteria, and they form spores during the production process. So that takes about 24 hours or so for those bacteria to sporulate. They can attach onto flock particles and do things to stay in the system, but it would take them about some time to wake up and start. When they're in the right environment, they can sense that environment, and then a whole process starts where they germinate from that spore and then become active bacteria in the treatment plant.
Okay.
I would say the nitrifying bacteria are, again, non-spore forming. Those are vegetative bacteria, so they will start working immediately when you put them in the system.
Let's see. Here's a question around how do I get a DNA test done and how much sample is needed? And I can answer that. Sure. Just feel free to reach out to your water treatment supplier or contact us directly. We're probably working with them. But you need 1,000 mils, and just reach out to us, and we can guide you through that process and coordinate with Aster Bio to get those samples done for you, and then plus help you review those results when we get the report.
Yeah.
And then looks like we got one more question. Let's see here. It looks, it's kind of related to the other one. How long will the bacteria stay in the system?
Yeah. So that's a good question. And I think that, again, is going to be a case-by-case basis. We've done some tracking of strains in the past and know that, some of them stay in the system and work for a long time. Some of the bacteria, nitrifying bacteria, we've applied them into systems where the nitrifying bacteria that we put in are not the bacteria that stay in the system for the long haul, but we can kind of use that augmentation for a couple days as the system recovers. So that's an application where those bacteria might not stay in the system for a long haul, but can be active in the system during that time, during that recovery while the system repairs itself. And sometimes they are the bacteria that predominate and stay in the system. But Greg, we're at 2 o'clock right now, so probably a good time to shut it down.
Yeah, no, thanks everybody for joining. Feel free to reach out to us with any questions. Let us help you troubleshoot. Everybody will get a copy of this presentation at the end. Again, thank you very much for showing up, and have a wonderful day.
Bye-bye.